Welcome back to Antibody Basics, Part 13. In this episode, we'll talk about therapeutic targets, specifically immune checkpoint inhibitors. Immune checkpoints are regulatory pathways in the immune system that modulate immune responses and maintain self-tolerance. They are crucial for preventing autoimmune reactions, but can be exploited by tumors to avoid immune destruction. These checkpoints act as brakes on the immune system, limiting the intensity and duration of the immune responses to minimize damage to normal tissues. Key checkpoints include the CTLA-4 and PD-1 checkpoints. CTLA-4 pathway is primarily expressed on activated T cells and regulatory T cells. It competes with CD28 for binding to B7 molecules on antigen-presenting cells. B7-CD28 interaction provides a co-stimulatory signal necessary for full T-cell activation. CTLA-4 engagement delivers an inhibitory signal, thus dampening T-cell activation and proliferation. The function of this pathway is to modulate the intensity of the initial T-cell response in lymphoid organs, preventing overactivation. On the other hand, the PD-1 pathway is expressed on T cells, B cells, and some myeloid cells. PD-1 interacts with its ligands PDL1, programmed death ligand 1, and PDL2 on tumor cells and APCs. Ligand binding delivers an inhibitory signal to the T cell, reducing its activity and promoting T cell exhaustion. This checkpoint functions at the effector phase in peripheral tissues and the tumor microenvironment. The function of the PD-1 pathway is to limit T cell activity to prevent tissue damage during inflammation and to maintain tolerance in the tumor microenvironment. Immune checkpoint inhibition is a mechanism for tumor immune evasion. In cancer patients, tumors often overexpress PD-L1, exploiting PD-1 on T cells to suppress immune responses. Tumors attract regulatory T cells and myeloid-derived suppressor cells that express checkpoint molecules like CTLA-4. The tumor microenvironment is enriched with factors that promote immune tolerance and inhibit effector T cell function. There are several ways to stop this, including restoring T cell function, blockade of CTLA-4 and PD-1/PD-L1 pathways can reinvigorate exhausted T cells, enhancing their ability to attack tumor cells, or tumor-specific responses, where checkpoint inhibitors can potentially re-establish a targeted immune response against tumor antigens, leading to tumor regression. Clinical trials have also shown that checkpoint inhibition can lead to long-lasting remissions in certain cancers, suggesting a sustained immune memory response. Other therapeutic strategies like checkpoint blockade, where monoclonal antibodies targeting pathways can disrupt inhibitory signals and restore T cell activation. This is effective particularly in cancers with high mutational burden, as it generates neoantigens that can be recognized by T cells, or an inflamed tumor microenvironment, where the presence of T cells and high expression of checkpoint ligands make them more susceptible to checkpoint blockade. An example of an anti-CTLA-4 therapy is ipilimumab, which is a monoclonal antibody approved to treat advanced melanoma, renal cell carcinoma, and others. It works by binding to CTLA-4, preventing its inhibitory signal, thereby enhancing T cell priming and proliferation. The drug was the first checkpoint inhibitor approved for melanoma, and ongoing research is exploring its efficacy with other therapies in a variety of cancers. Meanwhile, pembrolizumab is an anti-PD-1 humanized antibody approved to treat advanced melanoma, non-small cell lung cancer, head and neck cancer, and others. It demonstrated significant improvements in overall survival and progression-free survival in various cancers. Combination therapies involve combining immune checkpoint inhibitors with other treatments, such as chemotherapy, targeted therapy, or additional checkpoint inhibitors. This can enhance anti-tumor efficacy by attacking cancer through multiple mechanisms. Dual checkpoint inhibition, exemplified by nivolumab, anti-PD-1, and ipilimumab, anti-CTLA-4, which improved both T-cell priming and effector functions, leading to better survival outcomes in cancers like melanoma and renal cell carcinoma. Chemoimmunotherapy leverages chemotherapy-induced immunogenic cell death to boost checkpoint inhibitor effectiveness, as seen with pembrolizumab in non-small cell lung cancer. Combining checkpoint inhibitors with targeted therapies, like atezolizumab and VEGF inhibitors in renal cell carcinoma, modulates the tumor microenvironment and enhances immune responses. While combination therapies can achieve significant clinical benefits, they often come with increased immune-related adverse events, necessitating careful patient selection and optimizing of dosing regimens. Besides CTLA-4 and PD-1, there are other emerging checkpoint targets. For example, LAG-3, lymphocyte activation gene 3, is expressed on activated T cells, NK cells, or B cells. It binds to MHC class II molecules, inhibiting T cell activation and promoting immune suppression. Investigational therapies aim to enhance T cell responses in cancers resistant to PD-1, PD-L1 blockade. TIM3, T cell immunoglobulin and mucin domain 3, is expressed on T cells, NK cells, and dendritic cells. It binds to galectin-9 and other ligands, contributing to T cell exhaustion and immune evasion in tumors. Combined targeting with PD-1 inhibitors has shown potential for overcoming resistant mechanisms in cancers. TIGIT, T cell immunoreceptor with Ig and ITIM domains, are expressed on T cells and NK cells. This binds to CD155 and CD112, competing with co-stimulatory receptors and dampening T cell responses. Monoclonal antibodies targeting TIGIT are investigated to enhance anti-tumor and immunity in high ligand expression tumors. Biointron focuses on antibody discovery, expression, and optimization. From gene sequence to purified antibodies, it takes just 2 weeks. Our high-throughput platform enables the rapid expression of recombinant antibodies in HEK293 or CHO cells. We guarantee the delivery of 100 micrograms to 100 grams of recombinant antibodies. Biointron is also a well-recognized leader in VHH antibody discovery and production. With our self-owned alpaca breeding farm, we offer a 1 alpaca for 1 project commitment and guarantee the delivery of over 20 unique binders, high diversity, and large capacity. We also provide various services such as bispecific antibody production, afucosylated antibodies, single B-cell screening, hybridoma sequencing, antibody optimization, recombinant protein expression, CHOK1 stable cell line development, and much more. Thank you for watching, and comment with any questions you have below. Don't forget to like and subscribe for more.